Modern medicine is stalling against a specific class of illness. For decades, treatments for chronic, systemic conditions have hit a rigid ceiling of effectiveness. Conditions like long COVID, chronic fatigue, and widespread neurodevelopmental variations consistently resist standard therapeutic approaches. This failure stems directly from the linear biomedical model, the methodology that has dictated research and treatment for the last 75 years. This model assumes biological phenomena are produced by simple additive chains of cause and effect. A causes B, which leads to C. When a problem fits that shape, the model works. It isolated insulin. It built modern oncology. It solved the discrete mechanical failures of the 20th century. It struggles when faced with massive patient heterogeneity, when the same condition produces wildly different symptoms in different people. The linear model treats this individual variation as statistical noise, assuming the deviations are errors in measurement rather than meaningful data. The common response has been to gather larger datasets to average that noise away, searching for a single uniform pathway that doesn't exist. These systemic conditions reflect emergent properties of complex networks operating under intense constraint. Think of the linear model as a house. It has solid walls, clear light, and it functions for the problems inside it. But it is nested inside a much larger structure. That larger structure is governed by the rules of complex adaptive systems. In these networks, outcomes emerge from dynamic, unpredictable configurations of many interacting parts. Solving these modern crises requires mapping the holistic thermodynamic state of the body's entire regulatory architecture. Biological regulation is a physics problem of energy and organization. Chemistry is simply the language it uses to speak. This mathematical approach begins with Claude Shannon, who formalized information theory at Bell Labs. Shannon defined entropy strictly as a mathematical measure of uncertainty. High entropy means a system's state space is unconstrained and unpredictable. We can apply this measurement directly to human biological regulation, treating the body as a network of information streams. This chart shows how those streams coordinate. The first major data stream is the daily rhythm of cortisol production. The second stream is autonomic tone, measurable through heart rate variability. The third stream consists of inflammatory and immune signaling. These streams synchronize through a phenomenon called mutual information. Knowing the exact state of your cortisol rhythm allows an observer to reliably predict your immune signaling. In a healthy system, these correlation points represent coherence. The channels are informationally integrated. Under sustained demand, the system experiences stress incoherence. The correlation points break. The channels uncouple and drift into independent, chaotic patterns. This incoherence is a literal mathematical increase of Shannon entropy across the body. As the channels uncouple, the body begins visiting unconstrained, disorganized physical states that a coherent system would typically prevent. What medicine labels as a disorder is often the measurable thermodynamic degradation of mutual information across these regulatory channels. Ilya Prigozhin won a Nobel Prize for explaining how systems manage this entropy. He called them dissipative structures. This visual boundary represents the allostatic envelope. It marks the condition where an organism can successfully pay the energetic cost required to export entropy and maintain internal organization. When the energetic cost of maintenance exceeds the body's available capacity, the system reaches a bifurcation point. The internal links snap, and the network reorganizes into a high entropy configuration. This thermodynamic phase transition explains why clinical collapse often appears sudden rather than gradual. This framework clarifies why some distinct conditions appear similar under a microscope. Some clinicians currently argue that autism and Alzheimer's might be the same disease. They share metabolic signatures, such as high levels of oxidative stress. They both exhibit mitochondrial dysfunction and insulin resistance in the brain. The linear model sees these matching signatures and assumes a shared pathology, but it is confusing a shared metabolic substrate with a shared disease. Look at the regulatory architecture instead. Autism is characterized by a neuroperipheral configuration. This architecture acts as a high-gain antenna, processing significantly more raw sensory data than a neurotypical system. It also processes a higher volume of autonomic and interoceptive signals from inside the body. In terms of information theory, the system is operating in a higher dimensional state space. Coordinating these extra data channels creates a coherence tax. Maintaining mutual information across these extra dimensions requires a fundamentally higher baseline thermodynamic energy cost. Alzheimer's, by contrast, is the degenerative collapse of an architecture that has been run past its operating parameters for decades. Autism is a high-gain antenna calibrated for sensitivity. Alzheimer's is a scaffold falling down. The shared biochemistry reflects a shared energy infrastructure, not a shared cause. We can map these regulatory states topologically to move from symptom checking to systems thinking. The first dimension is metastability. This is the structural architecture, the existence of multiple accessible valleys that allow a person to shift between modes. The second dimension is regulatory bandwidth. This is the present tense energy reserves available to the person right now, within their current mode. This map dictates a logical sequence for support. Interventions aimed at restoring flexibility will fail if the system lacks the bandwidth to sustain the practice. Complexity evolution frames this high-gain variation through the lens of response diversity. Maintaining phenotypic variety within a population is a strategy known as bet hedging. It ensures some portion of the group is pre-adapted to survive. A species that optimizes for a single, efficient cognitive phenotype becomes catastrophically vulnerable to any stressor that phenotype cannot handle. Cooperative neurodiversity distributes the thermodynamic burden of adaptation. Some phenotypes maintain social cohesion, while high-gain phenotypes detect the signals the majority miss. The survival of the group depends on this distribution of regulatory strategies. Supporting these high-gain systems requires environmental design that honors their specific metabolic cost profiles. Forcing uniformity onto these architectures increases the mathematical fragility of the collective. Response diversity provides the substrate for adaptation. A population that protects its variation maintains its capacity to navigate a future it cannot yet predict.